I would like to kick off our fall forum officially with our first speaker, Professor Michael Thase of the University of Pennsylvania. Please join us in welcoming him.
Cool. So I'm delighted to be here with you. This is my second Philadelic and probably only my fourth or fifth talk ever involving psychedelics. So I'm a novice at this area of research. I'm not a novice at years lived and have some secondhand experience from the 1970, 1971 dorm life of roommates and people down the hall who did psychedelics. I was only the helper and observer and sometimes person who sat with the bad tripper. But it did provide some honest appraisal of the downside of what might happen with psychedelics not properly dosed and not properly supervised. My life's work has been the study of depression, and particularly difficult to treat depression. I'm a person who advocates academicians being involved with industry. I think the pharmaceutical industry represents the best hope to bring new, valuable, useful treatments to help those that aren't helped by our current treatments. I think they deserve the best advice possible. As you can see, I have many relationships. Today, talking about esketamine, you should know that I was involved with the development of that compound from about 2013 till ongoing now. That's once called Janssen and now called Johnson & Johnson. And then a company called GH Research, which is, I'll show you at the very end of my talk, which is developing a ultra short acting psychedelic compound that I am going to try to squeeze 15 minutes into the final couple minutes of my talk. It's honest to goodness truth that who you spend time with, who you earn money working with, influences what you think about, how you think about it, and so forth. The only way to mitigate this is to simply report what your relationships are and make it clear when you're talking about something that you've had some commercial experience with, that it's clear to the audience that you are not an unbiased source of this. So, in the depression arena, we've had really safer, easier to prescribe, newer versions of antidepressants since the late 1980s. Nevertheless, they didn't help any more people get better. They just made it a little safer and a little easier, safer for patients, easier for prescribers. These medicines are slow to work. They work inconsistently. They don't always treat the patient's most bothersome or troublesome symptoms, even when they are helpful. And you need 6 weeks, 8 weeks to fully evaluate the merits of a particular medicine if it's well tolerated. About 25% of depressed patients don't respond to any of the medicines we currently have available. Some of those will respond to other more advanced interventions, the most advanced other intervention, of course, being electroconvulsive therapy. I mean advanced in a sequence, not advanced in the science of it. The ability to induce a seizure electrically for therapeutic purpose was first discovered in the late 1940s. So just to make my point, this shows you drug placebo differences over the last 30, 40 years. And we have not gotten more effective medications. Placebo has gotten better over the last 30 years, and that's because the studies are done with more care and patients expect more in the studies. And so the drug placebo difference is actually a little smaller today than it was in the 1980s. The reason the antidepressants haven't gotten better is because they all target a certain particular set of mechanisms. And once you've not responded to one drug that does that, the chances that a slightly different drug that has the same basic targets will work is diminished. And by the time you get to 3 or 4 of those drugs, even as different as an SSRI and an MAOI, the chances that the patient will get better have dropped down to about 10%. And so this is why at the end of the day with pharmacotherapy, we get about 75% of the people who stick with us better. And just remember, each time a medicine doesn't work, there's a fresh chance the patient will just get frustrated with the process and drop out of treatment. We've needed new mechanisms to treat depression for years. This has been known since the 1990s that the SSRIs were a way station. They weren't the final solution to helping people with more difficult to treat depression. But mechanism after mechanism after mechanism that is theoretically relevant to the pathophysiology of depression has been tried, and sometimes they're not safe in humans, sometimes they just don't work. And the kind of mysteries of what depression is really remain largely unsolved at the level of our genetics, at the level our genetics interact with our life experiences and so forth. And you know, our first two antidepressants were discovered accidentally. One was a treatment for tuberculosis. The other was an attempt to make a safer version of Thorazine. And so this is serendipitous discovery. And the first new antidepressant in the modern era, ketamine, was discovered, its antidepressant effects discovered serendipitously also. Just to begin with the beginning, ketamine was a descendant of PCP, yes, the horse tranquilizer that is a terrible, terrible psychedelic compound. Ketamine, much safer than phencyclidine. Approved since 1970 in the United States. Ketamine cut its teeth as a new generation anesthetic in emergency rooms and in the Vietnam War in acute trauma. It is the one anesthetic that the World Health Organization said should be packed in an emergency kit of all the essential medications in the world. And it's the only anesthetic that a psychiatrist can learn to safely administer with our level of training. So that's a unique set of qualities. Of course, it's an abusable drug, and it's considered by the FDA Schedule III. It's not hard to prescribe a Schedule III drug. You simply need a standard license that permits you to do that. And it is sort of monitored at the state level, but you can have a lot of mischief. And much ketamine abuse involves university communities, people, dentists, anesthesiologists, vets, people who have not veterans, veterinarians. And there's just a lot of ketamine around. And plus, since it was introduced in 1970, it's amazingly cheap. You can buy powder and sterile water and mix it in the right concentration, and you can do it for less than 50 cents a dose. This is a remarkably powerful, inexpensive potential for, in our case, treatment of depression. I think importantly, this story winds through serendipity also, because this dissociative drug has some weak psychomimetic properties, and some healthy volunteers with family histories of schizophrenia participated in a study that was designed to help lead to new treatments that target the glutamatergic system. And they volunteered, they were given ketamine to have a sort of psychotomimetic experience to see if this could be blocked by a potentially new generation of antipsychotic treatments. It didn't work. The new medicine did not block that experience, but some handful of the original participants had personal histories of depression. They weren't screened to exclude based on their level of depression. And in debriefing, they told their investigators, you know, my depression lifted within hours of being in your experiment. And I stayed better for a week or 2 or 3 weeks. Now, the people who did the failed study then immediately did one of the smallest studies that you can do. Every participant was his or her own control. So it's a double-blind crossover study. Some got active ketamine first, some got placebo first. There were only 8 people in this study. 7 had major depressive disorder. 1 had bipolar depression. They hadn't responded so well to standard medications, but this wasn't exactly a study about treatment-resistant depression. And what I want you to notice here on the abscissa is that the x-axis, that you're talking about hours and days, not weeks, in terms of the onset of benefit. And now we're looking at the Hamilton Depression Rating Scale. Let's say the average person in a study like this scores 25. You can see, I hope you can see, that within 4 hours, the average person had a 50% benefit in their level of depression. And that level of benefit lasted for the 3-day follow-up that was done. Single dose of a sub-anesthetic dose of ketamine, only 1/4 of the minimum targeted dose for a dissociative anesthetic effect. So I was in the room at the Society of Biological Psychiatry in 1999 when this paper was presented. No one believed the results. I remember talking to colleagues the following year when the paper was published. Almost no one still believed the results. But there was a model to explain why this might have not just a large, but such an incredibly rapid onset of effect. And it involves the relevance of the excitatory neurotransmitter glutamate and its role in modulating fast-acting neural circuits. And so the idea was that at a sub-anesthetic dose, you could immediately switch down an unnecessarily pathologically activated circuit. And when you switch that down, it permitted kind of the normal processes of neuronal resilience to be facilitated. And so, it's easy when you have an observation of a powerful effect to think about a way that it might work. And then comes all the hard stuff like replication and then trying to bring something that's safe and reasonably effective to market. So there was no commercial interest in developing intravenous ketamine as an FDA-approved antidepressant. This is because the drug had been generic for 10 years already when this finding was observed. So there was no incentive to do that. But the world needed better antidepressants for people that didn't respond to the things that we had and as an alternative to ECT. And here are the first 5 replications. These are all small studies, some of them supported by federal funding, some supported by other funding, none supported by the pharmaceutical industry. Please look at the replicability of the findings, different continents, different investigators. The top plot is the 1-day effect. The bottom plot is the 1-week effect. Single dose, again, 1/4 of the dissociative anesthetic dose. So it's 0.5 milligrams per kilogram. So now the question is, we know it works, it's widely replicated. How does it work compared to a requisite treatment for people with real difficult to treat depression? Let's compare it to ECT. So these are a series of similarly small studies in which ECT and ketamine are randomized. Now, please remember, although ketamine is an anesthetic, it is not a good anesthetic for ECT because it is too light. It's a dissociative anesthetic. You don't get into the kind of deep state that you need to protect your bones from the electrically induced seizures. So you can't do ketamine as the anesthetic in a study of ECT. So in this trial, you've got a blinded evaluators in these trials, blinded evaluators, but people know if they're getting ECT or ketamine in these studies. This forest plot, if you've got good eyes, it doesn't show so well in this room, I think. The odds slightly favor ECT in this meta-analysis. The average subject in this series of studies has about an 8 to 10% better chance of responding to ECT than IV ketamine. Now, just a show of hands, who here, bad depression, nothing's working, who would want to have ECT versus who would want to have, let's say, 8 doses of ketamine? Could I have the ECT votes? So I would want ECT if I was delusional and I was incapacitated, because I would want the 10% advantage for that. Now, I wouldn't be able to work as a psychiatrist for at least 4 to 6 weeks, maybe longer because of the after effects of ECT. But there is a real difference in the person's experience that would mean even a modest replicable advantage for ECT doesn't mean that IV ketamine can't be an alternative. But let's look at the one large federally funded study in which you had outpatients, not inpatients, some people still working, average age in this study 10 years younger than the average age in the meta-analysis, and suddenly there's a 10% advantage for intravenous ketamine. Again, 0.5 milligrams per kilogram. The ECT people hate this study. They hate it because it's outpatient. They hate it because if you had psychosis, you were excluded from the study. They hate it because it was only 3 weeks long, so you could only have 9 ECT in 3 weeks. You got twice a week ketamine, so you got 6 ketamine treatments. Ketamine people don't hate this study, even though you normally do 8 ketamine treatments in a study, because of course you don't hate it when your side wins. So here we are now, 25 years after the serendipitous discovery. It is still, IV ketamine, still an off-label treatment. There are 1,000 clinics or so in the US. The reason I can't tell you how many is there's no central registry. And some clinics open and close and so forth. So the number could be anywhere between 800 and 1,200. I wouldn't know. That's my confidence interval about how many clinics there are. This is a medical intervention. It has to be done with monitoring of patients. Occasionally someone will have an anesthetic reaction, even though you only gave them 1/4 of the anesthetic dose. Many of us have our blood pressure go up and our pulse go up, and we really are disabling under the influence of the medication for 40, 50, 60 minutes around a treatment. Although there are protocols of how to do it safely and properly, there is no obligation to do it safely or properly, and there's no central overview of the procedure. So there's a lot of cowboy and cowboy girl-esque practice out there that deviates from what is recommended. Just to give you an example, there are places, if twice a week doesn't work, where you'll begin to get 3 times a week. Or if 0.5 doesn't work, you go to 0.6, 0.7, 0.8. Now, how does ketamine get you in trouble when you use it as a rave drug? Well, you take more of it more often. Okay, so all the safety data that has been so reassuring these 20 years does not apply to high dose, high frequency practice. And I had a patient come for me for a consultation who was having daily ketamine treatments, because that was, when you begin to have daily ketamine treatments, it's hard to separate therapeutic use from abuse. So it's hard to know, but that's where we are with the state of the art for ketamine. But what ketamine did was open a door, and that door led to the first pharmaceutical company expressing an interest in this area, and that was Johnson & Johnson. And they got the patent for the S enantiomer. How many of you have had organic chemistry? Okay. Some crystals in nature exist in forms that deflect light to the left and reflect light to the right. And these are mirror images of crystals. And I could show you, but I'm down to 10 minutes left in this talk. The S enantiomer of ketamine is slightly more potent at the 2010 target, the NMDA receptor. That's no longer why we think this is essential for working for the drug, but the patent was available. And so around about the time that first meta-analysis was published, J&J began an FDA-registered program, a proper program, and today there are 2,000 plus facilities. Now there was bad luck with timing, because the first year the drug was out came the pandemic. And the Janssen model was intranasal treatment. Now you remember we all wore masks. We were all scared to go to healthcare places during 2020, 2021. And so nobody really wanted to come in and get intranasal esketamine. The first couple years it was around. But it has now taken off, and in the past 15, 16 months, the use has doubled in the United States. So I won't ask who has personal use with shooting up versus snorting drugs, but generally taking something through an IV is faster and gives you more exposure than snorting something through your nose. And this is because your nasal mucosa is rich in blood supply, but not nearly as rich as your actual blood supply. And also, we have different thickness in our mucosa and different amounts of hair in our nose. So that the medicine can glob on just to the hair in your nose, and you don't trim all the hair out of your nose to do this treatment. So you get about 80%, I'll say, exposure with intranasal delivery. And because the company didn't want to make this too complicated, they made 3 doses, 28, 56, and 84. And 84 was what the 80 kilogram person would get, something close to 0.5 milligram per kilogram exposure. So now if you're 320 pounds, 84 is not getting you anywhere near that. If you are 98 pounds, it's getting you more than that exposure. So just like IV ketamine itself, the standard treatment is twice weekly for 4 weeks, or a total of 8 sessions. Yes, okay. And so consistency of effect, smaller effect than observed with IV ketamine, but consistent. Smaller in part because the design of these studies differed in the sense that every participant started a new antidepressant at the same time that they started either placebo or intranasal ketamine. It is a confound. You cannot really do meta-analysis since nobody starting IV ketamine starts a new antidepressant at the same time. But it is a consistent effect. J&J did do a study with no concomitant antidepressant. They demonstrated that the effect's actually slightly larger when you don't have an antidepressant on board. That's because the placebo group doesn't do as well in that scenario. And here we are 6 years out. The use is growing. There have been no red flags. And the REMS, the risk evaluation and mitigation strategy that the FDA has tied to this, basically eliminates mischief. It eliminates sourcing of the drug. Only the treatment facility has the drug. Adolescents cannot get into their parents' medicine cabinet and toot esketamine. The facility possesses the drug. You can't go above 84 milligrams. You can't dose more often than twice weekly with it. There are some annoying things about it, like if you need twice weekly to stay well, you still can't get that sometimes. You must make the transition from twice weekly to once weekly and so forth. So we know meta-analytically IV ketamine looks stronger than intranasal esketamine. We know there's artifact involved with that. But here's a comparison of intranasal esketamine against adding an antipsychotic to a patient's antidepressant. And you can see a pretty clear winner on both dependent measures, fairly rapid and fairly sustained across time. So that brings us to now how this kind of opened the door for the psychedelics. And as you know, that before 2010, there were no proper treatment studies done with psychedelics, with control group and informed consent and so forth. Circa 2010, with the recognition of ketamine's antidepressant effect, psilocybin was clearly in the best position of having an intermediate length to the experience, as well as the best safety data, honestly, based on recreational use and some studies of normal controls. And once the positive data for psilocybin started rolling in, now a number of other psychedelics, some related to psilocybin, some different, have begun to emerge. There is a belief, we know how these compounds work, and this belief is not much different than it was when I had psychopharm in the 1970s. The difference is we didn't know about the glutamatergic system the same way then. And indeed, serotonin 2A axons project and touch the same sites on GABA and glutamate that ketamine more directly activates. When you activate those sites, that does cause the blocking of an inhibitory effect. So now we've got all sorts of studies of all sorts of psychedelics going. I would need the hour version of this talk to do more about this, but I want to predict next year or early in 2027, psilocybin will be approved by the FDA. They're half the way there. The study that will be necessary for the FDA approve will be done later in 2026. I also want to spend a moment on the psychedelic that I've had the most contact with, the most research experience with, and that is mebufotenin, which is the synthetic version of Sonoran toad venom. And of course, the Sonoran toad blinds its predators briefly with this very rapid acting psychoactive substance. Its pharmaceutical name, mebufotenin, its manufacturer calls it GH001. And the study I'm going to show you a very 2-minute long snippet of is the first double-blind, properly controlled study of mebufotenin. It's an 8-day double-blind period followed by a 6-month follow-up. And during the follow-up, if your depression is not remitted, at the start of the follow-up, you can have active drug. And once a month, you're evaluated. And if your depression is relapsing, you can be redosed. And you can have up to 6 redoses in the study. Effect size 2, not 0.2, 2. There is no placebo response here. But remember, there was no placebo response in the first study of ketamine either. People weren't expecting to get better. They knew nothing about this drug. People got better in 2 hours, maximum improvement at 2 days from 1 day of dosing. I'll tell you more about dosing in 1 minute. And they stayed well for the 7 days after that. So I say 1 day of dosing because it's an individualized dosing regimen. 6 milligrams. If you don't get it at 6, you get 12. If you don't have a peak psychoactive experience, you go to 18. If you don't have it at 18, you're out of luck. So maybe there'll be another cohort, another time studied with even larger doses. Talk about replicability. On your left is the group that got it double blind. On your right are the placebo non-responders who on day 8 immediately qualified to receive the active drug openly. They had the identical magnitude and time course of experience, despite having not shown a whiff of improvement on the placebo. I guess I should tell you, you inhale this. It's not injected. Okay, it was a warm aerosolized inhaler in the original study. The new US version will be different. Here's what happens over the balance of the 6 months of follow-up. The average person who responded needed to take 3 additional doses over the 6 months. So this is not, 3 people had a miraculous rapid improvement and stayed well for 6 months. Most of the responders needed redosing ranging from 1 to 6 redoses. So I think I'm just a minute or 2 over, and thank you for indulging me, Peterson. So depression remains one of the world's great public health problems, and we have many treatments available, but there's a substantial subset of people who aren't helped by the treatments that we do have, or who don't want to have some of our more advanced treatments. The serendipitous discovery of intravenous ketamine led to the commercialization of intranasal esketamine, which opened the door for reconsidering psychedelics as a therapeutic tool. It's no coincidence that the first open-label modern studies of psilocybin occurred coincident with the reporting of the widely replicated findings of ketamine. It just took some prepared minds in England to do 2 studies of desperately ill people, open label, which led to then the new generation of studies that you've seen. This, in my time in the business here, which is over 40 years now, I've never seen such excitement and enthusiasm about what's ahead in terms of these evaluating these new mechanisms. And I think that does offer the best hope for our patients who aren't helped by the treatments that we do have. So see you a little bit for questions. Thank you.
I think only Dr. Thase could get through all the psychopharm of 4 different substances, future directions, what's going on right now in such brilliant detail in the 20 minutes that we gave you. That was brilliant. 22. I'd now like to turn the time over to the estimable Professor Del Rasames from Columbia University. Please give us a warm welcome for Professor Sames.
Thank you very much. Good morning. So great to be here. I would like to thank the organizers and the volunteers for having me and for including Ibogaine in the forum and including discussion on this molecule in a prominent way. We'll have clinicians talk about it later. So I would like to give you an overview about this molecule and some results from our own research, and also some theories, a new theory, how we conceptualize how this substance might actuate its remarkable effects. So let's get going. I will first get out of the way the financial conflict of interest, which we're required to do. So I'm at Columbia University is my academic part of the activities, but I'm also a co-founder and heavily involved with Gilgamesh Pharmaceutical, which is a startup, relatively new company, where I'm a co-founder and collaborator. We're also licensing partners, so some of the Iboga analogs were licensed from my lab to Gilgamesh. And as Dr. Thase touched on, there was nobody interested in these type of molecules, so there was no other way than to just start our own company as a vehicle towards clinical research. Okay, so this is a remarkable story, highly improbable story that starts with pygmies in the forest of West Central Africa, Gabon, and other countries, where it was used as a sacred medicine, and that spread to native tribes in those regions. And it really, Iboga, which is a short for the Iboga tree, or Tabernanta iboga, has been a central substance around which spiritual practices and religions were developed and practiced. So the history is really interesting, as the substance then moved to Europe through colonialism, and particularly French colonialism. And so there were these various periods of relatively intense research into Iboga, the material, the root bark of the tree, or isolated Ibogaine, so the isolation of Ibogaine and research around it. In fact, in France, it was a commercial product, lambarine, which was an extract from a related species and used as a stimulant, broadly speaking. So that's an interesting subchapter of this trail of discovery, as I refer to it. And then, of course, the seminal discovery, also serendipitous discovery by Howard Lotzo, who showed that it acts as a rapid interrupter of addiction, particularly opioid addiction. And then I have some names of major players that this medicine that's been used by pioneers for now decades, largely outside of the major medical system, or the main medical system, and basically in clinics set up all over the world, which of course Ibogaine, as the main psychoactive alkaloid, is still Schedule I substance in the US. And then some of the molecular insights, the question, how is it possible that one administration of this substance could interrupt dependence on opioids? Some of the molecular hypotheses were published trying to explain that, and that's where we entered the picture about 20 years ago, and we really started to focus intensely on this area, and that's our contribution, which I'll summarize today, particularly looking at mechanism and developing new analogs, dealing with the cardiac issues that I will talk about, and so on. And then, of course, other discoveries working with veterans, which are getting a lot of attention, showing that this molecule really will have much broader impact, even beyond the addiction, which on its own is remarkable. And then, of course, what's going on right now, the state initiatives and so forth. Okay, so the one major reason why Ibogaine really has been a niche within a niche, even within the psychedelic community, it was considered not really a psychedelic for a long time. The one major reason why it's getting traction and interest is, of course, this tragic reality that we're dealing with in the United States, and that's of course the drug overdose epidemic. The numbers are getting a little better, but still they're outrageously high, and not only the dependence on substances, but also the mental health crisis epidemic in general. So what has emerged, even in the early studies in Ibogaine clinics in Mexico and elsewhere, is that this substance is addressing multiple major hallmarks, if you will, of addiction or of substance use disorder. So it appears to address the dependence itself, and it mitigates acute withdrawal symptoms, but it also mitigates cravings. It interrupts maladaptive habits, and also addresses things like social anxiety and trauma. So it appears to be addressing key defining features of addiction. So in that sense, it appears unique and very powerful. This is just to show you what the data looks like. Again, these studies are open label, so basically everything with the exception of one clinical trial on cocaine dependence, all clinical data we have is either anecdotal or prospective studies or retrospective studies, surveys and things like that. So that's something to keep in mind, that from the classical point of view, from a rigorous medical point of view, these are preliminary. But still, when you see the numbers, right, there are hundreds of people treated by Deborah Mash when she had to do these studies, of course, outside of the US. You see large effects again at attenuating the withdrawal symptoms. This is just a quick summary, which I think gets to the point that at about 3 days post-treatment, you see effect that is similar to methadone. So it's as powerful as switching a patient from illicit opioids to a maintenance opioid. So it's a remarkable large effects that are reported. Now, particularly working with veterans that went to Ibogaine clinics to treat their addiction, another serendipitous discovery has been made, which is that Ibogaine treats also PTSD and potentially, as shown here, TBI, which is traumatic brain injury. So it's treating the alcohol use disorder, which is what these veterans would go to Mexico to treat, and then they would find that their symptoms of PTSD and TBI dramatically improved. And here again, what you see on the y-axis is the larger number, the worse the symptoms. This is an interesting scale they use. This is basically overall functioning or disability measure, so you can see the dramatic improvements in 1 day and then lasting for a month. This is a scale for PTSD. This is a scale for depression, and this is a scale for anxiety. So basically, this is not what we see, right, in psychiatry, with the exception of ketamine that we just talked about. A massive response with basically only one patient relapsing at one point. So these effect sizes that Dr. Thase talked about, this is enormous, right? Effect size larger than 2, but normally with SSRIs, you would see something like 0.2, 0.3. Okay, so what it appears that Ibogaine is really a transdiagnostic therapeutic, and so it interrupts addiction and substance use disorder, not just opioids, but also alcohol, nicotine, cocaine, and so forth. So that's remarkable on its own, and the data is strongest in this department. Depression and anxiety, post-traumatic stress disorder, and also traumatic brain injury. There are other indications, so we have signals as the trail of discovery. This is how it all began. We hear stories, anecdotes from individual patients, and this continues as more and more people are treated in the Ibogaine clinics. We hear stories about CTE, so that's a chronic traumatic encephalopathy, right? The Navy pilots, athletes, and so forth, repetitive brain injuries. Very difficult disorder. There are no treatment for that. We hear about the brain fog of long COVID. Now more and more cases of Parkinson's disease, MS, and others. And then we're really getting into the neurology area, where we hear about these neurodegenerative and other injury-related neurological disorders, where we hear anecdotal reports of efficacy. So that's right? Stay tuned, and the story really is evolving. This is a very interesting and fascinating part of Ibogaine and Iboga alkaloids that has to do with the subjective experiences of something that we refer to as psychedelic states. And with Ibogaine, people often refer to these as oneric states, or it's referred to as onerogen, a dreamlike, inducing dreamlike state. Now, we are collecting in my lab, headed by Dr. Lankry, we're collecting the reports, the subjective reports from people. We have hundreds of those now. And so we're looking for general themes. What is the landscape? What is the shape of that experience, which could be very intense and very long on Ibogaine? And I rephrase those in kind of a tech language because sometimes the reports are so bizarre or fascinating that it appears that we're dealing with a really interesting psychotechnology, if you will. So this is, of course, potentially contentious here because, right, every journey or every experience is different. But we may flip that instead of normalizing it or somehow standardizing it, we may also see it as being personalized. It induces a remarkable upgrade of internal, memory search engine, if you will, description of scanning of subconscious material, the experience of having a mental interface with the Iboga entity, so to speak. So there actually is communicating to subjects what it's doing. And then there are visions of very advanced neurotech devices such as time travel telephones and phone booth to deities and things like that. And then what is extremely interesting is the messages that subjects receive about the social context, where they are, how they belong into their social group, and the importance of those social bonds. So that is something that I would like to return to if we have time. So just a couple examples, right? This is from a trial, a study in Brazil, Dr. Schenberg, and one of the patients, right, it is saying, looked like a scene from the Matrix movie. So I asked what it is, and I get an answer, and this was funny. The answer was, it is your brain being organized. It re-edits your brain. It is like if it was a computer, right? I mean, the feelings go to the feelings folder because everything was shuffled. So then that kind of feeling that I'm being scanned and things are being moved, and a pretty remarkable stories. This is an incredible testimony, if you will, from an Iboga journey. And so here the participant is referring to as a remarkable agent for revealing suppressed memories, and it talks about that it's confident that it will become a generalized defragmentation and self-reformatting tool. So let's hold this thought because I want to come back to it at the very end if we have time. This notion of very powerful psychospiritual effects. Okay, so let's take this body of data and ask the question, how is it possible, or how would we conceptualize mechanistically, going all the way to molecular level, how this array of effects could be achieved? So let's start with this, which for this we have the most solid data, because you see that we have these effects that we scientifically ascribe to different systems, to different circuits. We have different explanatory models for opioid dependence is very molecular, biochemical, right? Talking about trauma, social anxiety is very psychiatric or very psychological. So it suggests that this molecular system is addressing multiple different systems. So that's what I mean by talking about different domains or different biological scales, which is basically what we as humanity and scientific community, we have broken down the reality, observable reality into segments. We break it down to systems that are a little easier to study. So we have molecular scale or molecular dimension where we talk about molecular structure, and this is a structure of Ibogaine, right? We call it a small molecule because it's relatively small in size to these large molecules. This is an NMDA receptor, which is an ion channel. You can see it's the cathedral-like structure, very large, and ketamine is this small little molecule that is binding into the channel of this large molecule. And ibogaine and ketamine share this quality that they actually bind into a similar place in this molecule. So what are molecules? Molecules are quantum objects, and in my view, they encode tons of information. And that information is released when they interface a living system. Also, these are static pictures, but in reality, molecules are moving and vibrating, so you can think of it as a dance, right? This molecule is dancing, and then the dance partner comes and it changes the tune and changes the rhythm of the dance, right? So and the information transfer is how that dance is transferred to another dance partner and another and another. So all that is in motion. So small molecules are informational codes. And then, of course, when we go up into different scales from molecule to cellular, so then we talk about cells and how cells work and how they interact. And then we talk about physiological circuits and organ level. And then we talk about organism level and then psychological, psychospiritual. So these are kind of these categories that we created for ourselves. So the hypothesis one here, global hypothesis, is that ibogaine somehow is operating across the biological scales. Okay, structurally, ibogaine is related to tryptamines. It is a very complex tryptamine. So it is a compact but very complex structure. And if you can see this unit here, if you deconstruct ibogaine, as we are doing in reality in the laboratory, you end up with 5-methoxy-DMT, which is the bufo psychedelic or toxin that we just heard about a few minutes ago. Or in reverse, if you construct and add complexity, you end up with ibogaine. So we as chemists play the Lego game. We put on different atoms, take off atoms. So this is this atomic Lego game that we play. We can actually create these molecules and then ask questions. Which part of the molecule is critical for this effect and so forth. Okay, so this is what we teach and have been teaching for a long time. Molecules like ketamine or other molecules interact with one target or very few targets. That changes the dance of that molecule. And that, of course, has downstream effects as the information propagates in the cell and so forth. Now, what is emerging that ibogaine does not operate through this simplistic pharmacological paradigm. So how does it work? So before we get there, I would like to conceptualize this further because this is extremely simplified picture. Each molecule, each receptor, enzyme, and so forth is, of course, embedded in a very complex matrix of interaction. So this is an artistic representation of what we're talking about. So here, this would be, let's say, the target that one target, one molecule, but it's surrounded. And so nodes mean molecules, enzymes, receptors. Connections mean functional connectivity. So information transfer, physical connectivity, functional connectivity. So for example, psilocybin or other molecules would act largely through one receptor. And so that would be within the classical paradigm. But that interaction with the receptor then propagates through the network. Now, if I use this as a representation of how psilocybin might work, this is how ibogaine would work. Okay, it's like an octopus, or rather a polypus, and it's just penetrating the entire living system and it's tweaking. Okay, it's tweaking throughout the matrix. So we call it a matrix pharmacology because we propose it is modulating throughout the living system. Okay, so I want to use a couple examples because we have a broad audience here with diverse experience and expertise. So what does this mean? So let's take a look at the traffic jam in Manhattan. And that's a daily occasion, right? And so then how would you solve, imagine that you have a jam, how would you solve this problem? So if you use the logic of classic pharmacology, you would go after a hub. Let's go into a traffic hub such as Times Square or somewhere around Madison Square Garden. And we're going to open streets, close streets, and do something which will then, of course, such an important hub, they will propagate throughout the whole network. Now, imagine if a technology where we would be modulating many different streets and lanes at the same time or in some sort of a prescribed sequence. Furthermore, what we see with Ibogaine, that it's modulating the critical pathways at multiple sites. So imagine Broadway here, it would be modulating at several different streets, but it's also modulating streets in the upward and inward. And that, so then it's some sort of a deeper logic there. If you're modulating so many different things with one goal, there must be some deeper logic. All right, so how about if you don't like traffic, let's talk about a global planetary information system. So now imagine a planetary disinformation crisis. Hard to imagine in these days, but it's theoretical, okay? So here's the lady, and she knows we're in big trouble, so she's doing her best. She's in New York City, and she's making phone calls, sending emails, and it propagates through the entire system. As an agonist, what we call an agonist in biology and chemistry, pharmacology, she turns on the computer and makes these calls. An antagonist would come, sit in her chair, and disallow her from making these calls, okay? So it's an agonist-antagonist. So she's making calls, she's making new transitions, she's blocking transitions, trying to solve the crisis, and they will, of course, propagate from a major hub throughout the system. Now, this is how Ibogaine works. It really is a totally different technology that modulates many different transitions, but also it's not necessarily starting or blocking, it tweaks the strength of these connections. So it increases the connection or decreases of different connections. If it works this way, then there must be a fundamental different and global logic, how you orchestrate this change. Okay, so back to the biology. So then how do we define Iboga matrix pharmacology? We were able to put this in print, so you can read about this in this preprint, but I'm just happy to report that it was accepted by a peer-reviewed journal, so this is actually now. Thank you very much. So proposing something slightly outlandish like this, it was a battle with the reviewers, but eventually we agreed that this is a useful theory. Okay, so Iboga matrix pharmacology is defined by numerous weak interactions. So there are many different interactions throughout the system, but they're weak, meaning they're not very potent. So again, it's very different from, let's say, LSD. So what is the logic? It appears there must be some sort of a molecular intelligence. Okay, so we're now solving the puzzle. Guided by this hypothesis, we're discovering, examining the pieces, the individual pieces of the matrix, and how does it all fit together? So let me give you a couple examples. So let's just dig a little deeper into the actual science. So for those of you who would like to see a little bit more concrete examples. So we discovered that Ibogaine and its metabolite, major metabolite noribogaine, is blocking this particular protein called VMAT, which stands for vesicular monoamine transporter 2. Critical protein for neurotransmission of monoamines, serotonin, dopamine, norepinephrine, histamine, because it packages the neurotransmitters in the synapse into the synaptic vesicles. So let's just zoom out. Imagine a neuron. This is one cell in the cortex, let's say, and its extensions, and a different neuron in blue, which comes from D part of the brain, serotonergic neuron, serotonin neuron, projects and connects onto a cortical neuron far away. And these dots are then synapses, connections. That's the functional atom of the brain, right? That's where most information exchange takes place, synapses. Synapses release chemicals, neurotransmitters, such as serotonin. That's neurotransmission. But to release the neurotransmitter, you have to package it first. And once it's packaged, vesicle fuses and releases serotonin. Noribogaine blocks selectively this VMAT protein. So that's a very important intervention. And we can study that quantitatively, so we can see, it's dose dependent. we've developed this fluorescent serotonin. So we have state-of-the-art new techniques that we can see this with spatial resolution. So we look at these events at individual synapses in the brain and so forth. And then we dig deeper and then we see, well, it's doing more than that. It's blocking it, but it also causes a reverse transport. So it's pushing some of the neurotransmitter out of the vesicle, but it works only partially. And when you look at tetrabenazine, which is a well-known blocker of VMAT, used to treat, it's actually an approved drug, right? Huntington disease drug. It doesn't compete with tetrabenazine, which is what this shows. So it suggests that, again, it's binding and it's interacting with this molecule in a unique, different way. Ibogaine is emerging as a molecular teacher. All right, but it's not doing just that, it's doing other things. So yeah, this is one thing I want to tell you before we get to the other molecules, is that VMAT2 blockers, such as tetrabenazine or reserpine, at sufficient doses induce catalepsy. So it leads to depletion of monoamine neurotransmitters, and you end up with cataleptic rabbits, mice, and humans. Catalepsy is a fascinating state where it's rigid postures are held. It's hard to make volitional movements. People are conscious, or animals, but they cannot move. So you can induce this reliably in mice. They stay on the bar where normally they would come off. And when we do this with noribogaine, which is a VMAT blocker, it doesn't induce catalepsy. And we dose and dose, and we go higher and higher in doses. There's no catalepsy. Okay, so it's an atypical VMAT blocker. So, but it also does the following. It also blocks SERT, which is a serotonin transporter, which is on the surface of the synapse or on the surface of serotonin neurons. But it doesn't only block it, SSRIs do the same, but it binds to a different site than SSRIs and induces completely different dance than SSRIs. And it functions as a pharmacochaperone. What is that? That's just the new thing, right? So it actually helps to fold the protein into the right shape. And there are mutations that cause folding deficient proteins, which can cause deadly disease. And Ibogaine can rescue a genetic mutation. It can actually help the string to fold into the functional protein. So this is what it looks like to be an Ibogaine researcher. This is bizarre, fun, and complex. It's like every day there's some, oh, there's this, there's that. And it's, wow, what is this? So now we have this picture that it's a blocker, but a different dance, right? So it's pharmacochaperone, and now it's blocking two critical transporters in the serotonin synapse. Two absolutely critical transporters for neurotransmission. It blocks both of them at modest potency. So look, when we just do an interim summary, we have dual inhibition of these two critical molecules, but it's not too strong, just a bit. We have pharmacochaperone, which we don't understand long-term effects of that. And we have VMAT2 partial releaser, which again is a relatively uncommon feature. Okay, so now this picture shows what I just told you with microanatomical reference, so we see where the molecules are. We also found that there's a potential modulation of these auxiliary transporters. And there's a literature, we haven't confirmed that yet, but there is a literature, there's a paper claiming that noribogaine potentiates signaling of serotonin receptors. So it's not an agonist, it's not an antagonist, it potentiates the pathways downstream of the receptor. It's getting complicated. Okay, you're an engineer, you say, I don't understand this biology stuff. Give it to me in schemes. All right, there's a scheme. SERT, right, is interacting with VMAT, auxiliary interaction with SERT. These are all transporters. You package neurotransmitter, exocytosis, release a neurotransmitter such as serotonin. That acts on receptor and signaling continues. And look what noribogaine is doing. Modulating SERT, modulating VMAT, potentially modulating auxiliary transporters, and it potentiates serotonin. It's a very intelligent molecular octopus, right? It's going in and tweaking these pathways in a completely new way. All right, so this is a little bit of a look into what it looks like. And now let's just summarize this and organize our thoughts. So there's scientific data. So we are anchoring our research in the scientific method and try to be as rigorous as possible. So that's the data that look like this. Then, of course, we interpret the data as everybody else. So we show, okay, this is interacting with multiple targets. It's doing something very special. And then we extrapolate. Still scientific data, right? Still anchored in scientific data. What does this mean on a larger system level? And then we let the imagination and intuition work. So this, of course, is not typically presented at scientific conferences because you could be discredited very quickly if you come up with something too outrageous. But here we'll do that because it's a Philadelic forum, right? So this means that there must be this large logic, and this we know, and we intuit, imagine, and extrapolate that this is just a little piece of the whole matrix. This is just a little insight, little look inside under the systems like, but this is going to be a theme that will grow. That's our prediction. All right, so of course, what's the hook? So Ibogaine, as remarkable as it appears, does have serious potential adverse effects. It increases the risk of arrhythmia, cardiac arrhythmia. And there has been rare, but there has been deaths. This is an example of an ER room recording. You can see a very scary looking and life-threatening arrhythmia. So how do we deal with that? One is safety protocols. So that's what the clinics, Ibogaine clinics, do right now. And another solution would be, let's develop new analogs and deal with this cardiac risk. So question number one, can we develop safer Ibogaine analogs? Question number two, do these safer analogs keep the matrix? Do they keep the special sauce? They're going to be still active. With such complex pharmacology, you can imagine that tweaking that structure, we may lose that special activity. So that's two critical questions that we're really addressing in my laboratory. So we are chemists. So we make Ibogaine natural metabolites. There are other alkaloids in the Iboga plant. So we study them very intensely. So we, for example, we never use a Tabernanta Iboga, which is threatened. So we, for example, use Wakanga africana, so you can make from the seeds and the bark, you can make voacangine, which is related alkaloid, and then using special chemistry methods, we can make nor-ibogaine and ibogaine. These methods, of course, it's like specialized form of cooking. So there's a lot of heating and cooling and smoke and all that. And so each atom that you add or take off the molecule, you do it through some sort of a cooking. And there are a lot of catalysts and reagents that you have to use to actuate that specific molecular change. So that's atomic precision Lego game. Or we do fully synthetic construction of Iboga alkaloids. So we start with little pieces that are commercially available, put them together, and make the Ibogaine or Iboga analogs synthetically. Okay? With that ability, we can now essentially make any structural change conceivable. So including changing this nitrogen to oxygen or to other atom, we cannot do that from natural products. We don't have chemistry to do that, so we have to start from scratch. Okay, and so this is first answer to the question, can we make safer analogs? This is what we call oxa-ibogaine and oxa-nor-ibogaine, this molecule that has an oxygen instead of nitrogen. So seemingly it's very small change, structural change, which led to pretty profound effects. And I'll show you here, we use donor hearts. So these are actually hearts donated by victims of accidents and overdoses and things like that. And we use the cardiac tissue or dissociated tissue into cardiomyocytes, which are activated by electrical fields. So you basically have a mini heart beating, and you can see a pretty nice regular beat. With nor-ibogaine or ibogaine, we recapitulated ex vivo, meaning outside of the living human. We recapitulated the proarrhythmic risk. You can see it, even a non-expert, even a non-cardiologist like myself, you can see missing beats after beats. This is a scary looking profile, and that is dose dependent. So as we go up in concentration of Ibogaine, we have more of these events, which again clearly shows a pro-arrhythmic risk. And to our great surprise, when we look at the OXA, there was no such effects on pro-arrhythmia. So now we have multiple different solutions to this, and we're learning how to achieve this. But it looks like we are able to generate Iboga analogs that lack the specific risk of arrhythmia. The second question is, does this new molecule maintain the activity of Ibogaine? So of course, we have to model addiction and other conditions in animals, which is very hard, but nevertheless, rats are trained to self-administer with some work, heroin or other opioids, and then they're stably self-administering heroin. One injection of oxon or Ibogaine can lead to lasting suppression of Iboga. And we compare it to Ibogaine. So we're basically matching and sometimes outperforming Ibogaine in these tests, okay? So it looks like this might work. This is another example. This is a massive preclinical trial. It was several sites in Germany, outbred rats, so they're genetically more diverse. And you go through a year of inducing the alcoholism-like phenotype. So drinking, abstinence, drinking, abstinence. And this goes on and on, mimicking, right, the life of an alcoholic. And what you see is this abstinence effect, which is well known in humans, that after abstinence, the drinking goes up in humans and in rats. And our OXA compound blocks that abstinent effect. Again, matching and sometimes outperforming Iboga. So these type of experiments are being done to look at preclinically, does the new molecule maintain the qualities of the therapeutic-like qualities of Ibogaine? Okay, so what we're talking about now, that we are creating not just Ibogaine or nor-Ibogaine, but we're creating the whole new category of Iboga compounds, of Iboga therapeutics. I refer to them as molecular technologies. That's a linguistic transgression, but I feel that the Iboga demands that. It's just too sophisticated, too complex to call it a drug. So we're now in the imagination and intuition category on that spectrum, okay? So what I really see here is that we have a system, molecular system, that somehow reads, in quotes, through the scales, through those domains of life, from molecular all the way to psychospiritual. Somehow is reading it and tweaking it and changing it. So I see these molecules as antibiotics for neurological, mental, and psychospiritual health. This is, I think, a major break in medicine and in science. So this is what this arrow is trying to capture that we are penetrating through all these layers, molecular, physiological, individual, social groups, and so forth. So how about this? Now we're definitely in the intuition and the imagination realm, okay? So I just want to qualify that. We will need AI to understand Iboga pharmacology and Iboga effects, for sure. It's so complex. We will need that. We need that. We already are tinkering. So that's given. So that's this flow. We're going to use and develop new AI systems that will get deeper and deeper. But what if we get so deep that we will then turn the tables? We will use the Iboga logic to build a completely new AI systems. And here, crazy idea, but we are going from molecular algorithms to virtual. So I think we're closer to virtual than just observing patients and reporting in scales what's going on. This is really a long-term vision. We're trying to learn the logic of Iboga as a general corrective system that can read through the scales of living systems and bring it to the virtual, and hence save the world, of course. So let's just dream a little more. Okay, so we really want, this is now our goal. We want to decipher the Iboga molecular codes. We may never get there, but we will get deeper and deeper and deeper. And we will generate generations of therapeutics and psychosocial technologies. Addiction, psychiatry, brain injury, dementia. Very important. How about these inducible wakeful dreaming states? Unique mental states, personality reformatting, health span, and so forth. How many people are talking about algorithms to enhance human wisdom? Personal algorithm for searching for personal meaning and purpose, collective purpose. These things we need to think about. This is where Iboga is leading us to. I get goosebumps. Now you think, okay, so step one, so we're fully aligned with the Americans for Ibogaine, and Brian Hubbard will speak today with what's going on. It's remarkable, right? $50 million from Texas, $5 million from Arizona State, and other states are joining. We need this to happen, to bring Ibogaine itself to safe use, a legal use in the United States. Step number two, we need the Iboga Human Project. We have Human Genome Project. We have Human Immunology Project. It's a consortium that has $8 billion in budget. We need Iboga Human Project. So we need $1 to $2 billion to start with. Think about that. Help us to make it happen. And if you think that I'm crazy, then let's look at this. Remember that one report from a user saying that Iboga to her seems like a generalized defragmentation and reformatting, personal reformatting technology. Now, when I reread the old masters, so these are ethnographers, okay? Ethnobotanists, Ballandier, French, spending time in Gabon in the jungle. This is one of his conclusions of his studies. Iboga demands a complete transformation of self, a liberation from a standardized and difficult existence. It restores order and confidence where the forces of disorientation and hence insecurity have flourished. That sounds like something we really need. Fernandez and Fernandez, ethnobotanists, Americans, they write, for Iboga there, Gabon, is regarded as crucial to the management of space and time and generational relationships. It affects a time and space binding of social experience in the broadest sense, right? Bringing that all together, the arc of humanity, where do I belong, where do we belong, and so forth. Okay, thanks to all these friends and people who paid. We, of course, when NIH turned off the faucet completely overnight, we were facing the cliff that we will have to close down our Iboga program. So philanthropists and friends stepped in and kept us going, which really is remarkable, including all these foundations and individuals. Mather's Foundation that kept us going for a long time. And indeed, when the faucet was reopened and when the government reopened, the NIDA is actually supporting our analog program. So thank you very much.
I'd like to introduce our next speaker, Paul Rogier. Paul, good luck, my friend.
Right. Make sure working. All right. Thank you so much for the invitation to be here and to talk about psychedelics and mental health, their promise, and some of the neural mechanisms. I'm honored to be among these esteemed presenters, and that was a very inspiring talk by Seamus and Dr. Thase, who we're collaborating with. So I'm not talking about our specific research that we're doing. Anna Rose is going to be talking about that next, our design. But this is really forming the conceptual foundation for why we're doing what we're doing and why we think cognitive flexibility is so important, and why it's changing, or what the mechanisms are of cognitive flexibility. All right, so mental health, still an issue. About 1 in 5 people in the United States, so this is United States centric, I apologize, but so 1 in 5 have some form of mental health issue. About a fourth of those are considered serious mental health issues. But the main point of why I'm bringing this up is that about 50% of these mental health issues are developing before the age of 14. And 75% are developing by the age of 24. So by the time our brains have matured, which occurs in the mid-20s, and you can see this here. I won't go through everything because of time, but as the brain matures, oops, sorry about that. Okay. So these mental health issues are developing alongside the brain maturing. So once the brain is fully matured, a lot of these mental health issues have set in. So at that point, the brain fully matured, it's very difficult to change these disorders. Addiction too starts early. And the most prevalent types of disorders are found in 18 to 25. So a similar issue that these are developing early, and by the time the brain is developed, many people have these mental health issues and addiction. And just to point out too, an interesting little tidbit about licit and semi-licit substances is that the licit and semi-licit substances tend to have more of a mild or moderate sort of substance use disorder, whereas the more illicit or illegal substances tend to be more severe. And that doesn't have to do just with the chemical processes that are occurring or the biological processes, but also the way that people are using these substances. So it's something to consider as we need to intervene with these disorders. So some of the issues, though, is that when people have these disorders, many people don't seek out treatment. So the highest percentage of people seeking out treatment seems to be depression, but anxiety, substance use disorder, PTSD, these are all very low. So people may have some form of disorder, but they're not going to seek out treatment. And even when they do seek out treatment, within the first 6 months, we see retention drop by about 50%. So we may be getting some people in for treatment, but we're not able to keep them. And it drops a little bit more by the end of the first year, but really we're losing people within the first 3 to 6 months. So that's a problem. And so just for one example, and this is an example for first-line treatments for depression and subsequent responses in the first year. So let's say we have 10 people that are seeking out treatment for depression. 6 of them will, or we have 10 people who have depression, 6 of them are going to seek out treatment. Of those 6, 3, and this is all based on literature, 3 of them may go on to be responders, where 3 of them are going to be non-responders. Responder is just someone who has a significant decrease in their symptoms. So the non-responders are going back to square one and they have to start over again. And of those responders, maybe 1 or 2 will go on to be remitters, and 1 or 2 will relapse. So by the end of this cycle, within this first year, we have 1 in 10, which, we could do better. And so the other issue is that even if you have someone who is remitting or has gone into remission for their disorder, may experience tolerance. So after a while, these medications won't work as well. You might have to increase the dose, or you have to add on another medication, and you might start that cycle over again. Or someone just stops taking medication because they feel better or there's negative side effects. So this is a vicious cycle that people get into, and it's difficult to get out. So what is the promise for psychedelics? We've seen some of this already. But the promise for psychedelics is that they act quickly within a matter of days, as opposed to weeks for traditional psychotherapies. And they last a long time too. So even if you have 1, 2, or 3 treatments, these are lasting for up to months, sometimes longer than months. And so that's one of the main promises. This is the thing that is showing us that it may be something that is going to be revolutionary. But we're still in the early stages. So this is why it's psychedelic promise, but not a magic bullet. So in one slide, here's what the psychedelic promise looks like in the early stages. So the thing to pay attention to is the red dotted line. That's where the traditional benchmark is, where your traditional SSRIs or anti-anxiety medications or what have you are working as a % improvement above the comparator. So that could be placebo or some other comparator. And these are grouped by category. So there's depression, anxiety, substance use disorder, anorexia, even they're looking at. And then in the trials we've seen so far is that psychedelics are improving almost twofold above traditional benchmarks. And one to point out is this Psy-MDD, where they are comparing to a traditional SSRI, and it's showing about the same benefit that SSRI would show above placebo. So that's very promising as well. All right, so how does it work? I'm going to use psilocybin as an example. So the primary metabolite, the active ingredient of psilocybin is psilocin. It's a receptor agonist, so it's working on 5-HT2A receptors primarily. That's the most important receptor. It does have many other effects too. It's acting and binding on other receptors, serotonin receptors, there's a ton of them, but not as strongly. And so I'm highlighting the 5-HT2A receptor in red and the 5-HT1A receptor in blue. The 5-HT2A receptor is excitatory, so when it acts on a receptor, it activates that receptor and activates that neuron. Where the 5-HT1A receptor is inhibitory, and those are the receptors that traditional antidepressants are typically working on at 5-HT1A. And we know that 5-HT2A receptor is responsible for the psychedelic effects because 5-HT2A antagonists block the psychedelic effects. And we see a very strong correlation between the intensity rating of the subjective effects on the y-axis and occupancy of 5-HT2A receptors on the x-axis. So this is an interesting paper about these two different receptors by Robin Carhart Harris and David Nutt, tale of two receptors. So why is it important that it's acting on 5-HT2A receptors versus 1A? 5-HT1A receptors are primarily densely found in subcortical regions. And so this is something that's working, like Dr. Thase was talking about, over a long period of time. And oftentimes it's working at the subconscious level. So you're not even really processing that this is happening. And you may have taken antidepressants yourself, I have, but you sometimes don't even notice the difference until someone comes up and says, oh, you seem better. So these are working to decrease limbic responsivity, decrease anxiety, aggression, and overall help depression and well-being. Where 5-HT2A receptors are primarily found in the cortical regions. This is increasing 5-H signaling in those areas and encouraging cortical entropy, decreasing rigid thinking, increasing neuroplasticity. And this is something that we see both in 5-HT1A and 2A, where neuroplasticity is occurring in both, but it's much more limited in the traditional medications, whereas it's very extensive in the 5-HT2A receptor action. But both work to decrease depression, but just in different ways. And so when these are activated in these cortical areas, we have this affectation of brain networks. And so in a normal brain, these different colors are representing different networks, large-scale networks in the brain. And so typically, the networks are talking more within themselves. So they're more integrated within themselves. Whereas on psilocybin, you'll see more crosstalk. So this region's talking to this region over here. This region's talking to this region over here. So there's more exploration of these brain states. It's more entropic, more chaotic, and lots of less constrained exploration of states. So the global integration is increased because typically as we age and normal brains, our networks become more modulatory, so they're less talking to to other areas. And this allows the brain to sort of explore these states, but unconstrained. So the training, the preparation, the things that we're doing going into these sessions is highly important. So in this study, this is using mice, and I won't go into the tasks, but if you're interested, I'll talk about it later. The essential point is that there's a critical period for social conditioning in mice occurs within the first 21 to 49 days, that roughly equivalent to going into adolescence. But as it gets older into adulthood around age 91 days, it doesn't have any preference anymore for for a social setting. So social conditioning doesn't cause them to want to be in more of a social situation. However, if we use NMDA, psilocybin, LSD, ketamine, Ibogaine, all of these are reopening in a way these critical periods and allowing social conditioning to occur again. And this paper, again, it's preclinical, so this is just a model of what's occurring. But it appears that these critical periods are opening in a lasting way that's correlated with the duration of the subjective effects. So something like ketamine is very short acting, so that window that's open is only a few days. Psilocybin works for a little bit longer, so that critical period is open even longer, allowing a person to have to make more change over those days and weeks. So the other question, and I'll end with this real quickly, is that does the subjective experience matter? So if all this stuff is happening in the brain, do we even need to experience what is occurring? Do we need to have this experience at all? The three that I want to talk about are mystical experiences, which is a profound spiritual, strong spiritual experience. People talk about ego dissolution and oneness, openness with the universe. Psychological insight is ability to clearly see the problem. So these are like your aha or eureka moments. Alcoholics Anonymous would call this a moment of clarity. And psychological flexibility, which is the willingness to accept that problem and change, to actually make the change happen. So what we see is that psychedelics increase mystical experiences, and those who are responders or those who have higher mystical experience scores tend to do better in clinical outcomes. So clinical outcomes are improved. And we see this in smoking, depression, and anxiety. Psychedelics also increase psychological insight with higher doses, and higher psychological insight is associated with better outcomes generally. And then psychological flexibility is also increased. So this study is showing a decrease in psychological inflexibility, and that inflexibility, the decrease in inflexibility was associated with better outcomes as well. So it looks like that psychedelics are increasing all these different subjective experiences, and all these subjective experiences are associated with better clinical outcomes. But this paper by the Johns Hopkins group, they found that even though mystical effects and insight are predicting clinical outcomes, if you account for psychological flexibility and use it as a mediator, it's completely mediating the relationship. So it looks possibly, this is one theory, that it looks more like this, where accounting for psychological flexibility means that mystical experiences and psychological insight are no longer associated with clinical outcomes. Psychological flexibility is completely mediating that. So to me, that sort of means that you can have this profound experience and you can have insight into your problem, whatever that is, whatever you're working out with your therapist. But you need that psychological flexibility. You need that willingness to adapt and change for outcomes to actually occur. So it's really important for set and setting, I think, in that context, so that you have preparation that is guiding you for what is going to happen, and for some goals and ideas on what you want to focus on, and that you can make happen in the following weeks. So, just as a quick summary. The people with mental health disorders who get stuck in this vicious cycle may need extra help if they're in treatment-resistant depression or some other form. They may need a little extra help, and that psychedelics are a promising treatment for these disorders, that this is increasing global network integration and reducing habitual thoughts, behaviors by lessening entrenched networks. I didn't talk about entrenched networks. But I can more if people are interested. And we're seeing that they may open up a critical period. So things that establish early in life, like I was talking about with mental health and addiction, we may be able to open those up again and retrain those, recondition those entrenched networks that have occurred. And that my thought is that psychological flexibility may mediate all these effects of the subjective experience and clinical outcomes. And so in that way, I think that the subjective experience is a necessary part of treatment. So that's all I have. Thank you.
So when I was learning all about mood disorders, it took us about 2 weeks to cover what Paul was able to cover in the course of like 5 minutes. And they didn't make it in any way relevant and important and impactful as Paul did with the next 5 minutes. That was incredible. Thank you. Next up, we will hear from another one of our UPenn faculty, Professor Anna Rose Childress. And switching to a little bit more the translational side of things, we're going to talk about the challenges of actually doing a lot of this research and going from the rodent models to human models in some studies that are happening right here, right now at Penn. And she'll be tuning in virtually for us. Let's please welcome her, Dr. Childress.
Thanks, Peterson. And welcome to all of you. My thanks to Peterson and the organizers. I now have to follow these excellent talks as the closer for this early morning session. And here I am with Dr. Shamish removing long-time chemical barrier to Ibogaine studies. And here I am complaining that even after we get drug, we can face barriers to psychedelic research. That's hardly in the same league, but I hope it might be of use. My overall goal is very briefly, and everything's going to be brief this morning, introduce our team, our new psilocybin research, and then with some examples of the local barriers that popped up that we faced in getting these studies underway. And I hope this might be a help to others who are thinking about getting started on research, and also because you might have some good additional suggestions for us. Let me advance to the next slide. Okay. All right. So this is our research group here in the Department of Psychiatry in Penn in collaboration with teams at Drexel and Westchester. We were fortunate to receive in 2024 funding for psilocybin research in opioid use disorder, both for a clinical trial and a brain behavioral mechanism study. So that's the good news, and we're really gratified, especially as members of our team across the top row there, Dr. Regier and Gwiziak, have been hoping to test this medication in addictions for many years. In fact, in 2015, when Paul joined the lab, he asked if a 5-HT2A medication category could offer a research path forward for him. Now, at the time, there was almost no research funding for psychedelics. So I agreed that the 5-HT2A target was really intriguing, it deserved study, but I just couldn't see a way forward. But when the field began to break open a couple of years ago, thanks to many efforts of those in the room, we quickly moved to submit grants. And so the good news is we have some psilocybin grant funding, and we have a large and amazing multidisciplinary team. These individuals are really critical to doing the work, and though the time is really short, I want to quickly acknowledge them. So Kyle Campman and Michael Thase, whom you've already met this morning, are study physicians with deep experience in medication trials. Dr. Hassan Ayaz is our FNIRS imaging expert and a lead investigator at Drexel. You've already heard Paul, and Mike Gwiziak is our lead facilitator for psychedelic-assisted psychotherapy, PAT. Nathan Hager, also there on the top row, is a clinical psychologist, a 90T32 postdoc, and a co-facilitator for PAT. Emily Hartwell there on the bottom line and Claudia Gogowski on the second line are co-facilitators for PAT. Megan Ivy on that same second row is our highly experienced project manager. Altona Tufanoglu is our biomedical engineer who enables our task programming and does our analyses also for FNIRS and fMRI. Conrad Burns, who's also our timer this morning, and Sarah Benedict Augustine are research coordinators, and Cecile Denis is helping with her expertise in community involvement for recruitment. Melody Mendoza is our CRNP, and Chris Petro is our technical specialist. Robin Carhart Harris and Chris Nicholas from the University of Wisconsin are our deeply experienced with psychedelic research consultants. I'm being asked to start my video, and I thought I was already screen sharing, but let me know if this is not working. I'm going to talk with you now about our study design. That team that you saw enabled us to get funding, and the study design that we have here is basically a simple single dose, single day design with one of two doses of psilocybin, the low comparator dose of 1 milligram or 25 milligrams. And that's on the dosing day that you can see here in the middle. Prior to that dosing day, in the day's run-up, we have very important preparation sessions by the PAT team. We have a battery of brain and behavioral measures using FNIRS and neurobehavioral tasks, including neurocognitive flexibility, which Paul has mentioned. And following dosing day, we actually have integration sessions by that same PAT team. And about a week later, we have our post-psilocybin measures, another battery of brain and behavioral tasks. And we continue to follow patients in the clinical trial for twice-a-week urines. So basically, we have for the 8-week clinical trial, twice-a-week urines for 8 weeks, and we continue to check in with people over the subsequent year. We basically have, at this point, one barrier that you run into when you first start working with psilocybin is that there is no domestic provider really available. There's very few. And our first one, the negotiations with our university fell apart when they couldn't reach an agreement on language around intellectual property and indemnification. But the good news is that NIDA is now actually trying to make arrangements for a domestic central source of psilocybin for their grantees. But this wasn't in place at the time of the awards. So even before you can actually submit your protocol, if you're going to run off and do some research, you need to have a source. You need to have the investigator's brochure to give to your IRB to get started. Okay, so the good news is we got the medication. We had a design, but we ran into some barriers that we really hadn't expected, and that's where I'd like to spend the remaining time. Now, we know that all this is going to get better as there is federal rescheduling of psilocybin. We're looking forward to that. But of course, we want to do some research while these efforts are ongoing. So I'd like to tell you a little bit about the local barriers that we've bumped into. And also, get your thoughts on ways that we might be able to work around these shared experiences. So I've divided these into categories based on what I think is the barrier, and then how we think it was expressed or how we bumped into it, and then basically potential workarounds or potential solutions. So the first barrier is a familiar one. It's fear of the unknown, fear of something novel. For our university and probably most universities, these studies are very new. Our university had not seen a previous protocol with psilocybin, so they had no prior experience. They had basically reviewers who were doing their best. These are people in an academic uh, IRB, Human Subjects Committee, who basically are doing this on a volunteer basis. So they have specialties, but not necessarily in the area of psychedelics at all. In fact, not even a psychiatrist in this case, or even an addictions researcher. So we had, I think it was a cardiologist and probably an epidemiologist who were the primary reviewers. So this is difficult because the only thing they really did have to go on was the draft guidance that FDA is trying to work through for how to do trials with psilocybin and psychedelics. And as you know, that guidance is emerging and it's certainly not final. And some of the things in there are really sort of probably not what you would even recommend clinically. For example, the FDA guidance mentions, wouldn't it be nice for you to have a different person at integration than the person who has been the facilitator during dosing day? Because that looks on paper like it would reduce the impact of expectancies and biases. However, it's also not such a great thing from a safety perspective because you really want to make use of the fact that you've just developed this trusting relationship with the patient during the preparation and dosing day. So they were doing their best, but it was difficult for us. They had about 29 stipulations in the first round of review, and then it was really another 29 for the second round of review. It was very extended. And they ended up trying to think about our study as having safety elements, even though we were very emphatic that this was a phase 2A early efficacy trial, because safety, as has been outlined by other speakers, has really been very well established for psilocybin. But we ended up agreeing, because we had to essentially doing safety checks over the year following, for example, the 8-week clinical trial, knowing that much of what you'll pick up in those long time frames has little to do with the safety of one dose of psilocybin on one day, many, many months prior. And the reviewers, because they were reviewing the FDA guidance, also felt that they were rediscovering the challenges of blinding and expectancies and how to evaluate the role of the therapeutic aspects in psychedelic research. These are things that are familiar to all the people in this room, but basically they were rediscovering it and pointing out the challenges. So we had to sort of answer those things all over again and sort of renegotiate that with them, telling them that we weren't going to redesign the study to add another arm with psychotherapy only. We didn't have resources for that, and that wasn't really the point. We were trying to look for an early signal and seeing then about unpackaging designs as a secondary goal. So fear of the unknown, inexperience, and basically, at this point, a lack of people in the field who are able to have enough background in any of these domains, in addiction, in psychedelics, to sit on these committees. So one of the things that might be terrific is if some of the people in this room would love to volunteer to help with those kinds of reviews or getting our universities to accept that. Alternatively, it'd be great if the universities would have a professional IRB that basically could pull in people and essentially pay them to review so that expedited review would really be expedited review. We spent many, many months in this very early phase of people getting used to the idea of a psilocybin protocol being proposed. Ours was the first in our university for sure. And basically, we were the trailblazers, but we sort of have a blaze across our head from this. Second barrier of sorts is that there's still some stigma attached to studying psychedelics because, of course, if they've had recreational use, if any of them have, but psilocybin has, then there's concern that somehow your study is going to end up having a negative impact on the individual. Even though we're very clear that we go about screening the patients very carefully that are candidates, and that people who have had extensive psychedelic use or look like they were overusing, those are people that we would not be including in this study. Nonetheless, the human subjects folks will say things like they would like you to have some measures of potential abuse potential. They're worried that the people may have a rewarding experience. This is really sort of a catch-22, isn't it? Because you really would like for some of our patients to have what they consider to be a positive experience, not just a challenging one, but a positive one. But both FDA and our IRBs are struggling with this. And how do you evaluate how the people feel about the experience that they had? And they're worried that if anything is too positive, that it could be problematic. This is sort of flying in the face of what was mentioned in some of the earlier speakers. We know that you can have a drug that has a positive effect on the individual and actually still have great clinical benefit, but there is this stigma thing that we're still dealing with. So I think the main thing is to keep sharing empirical data about psilocybin's low abuse liability, but also just help the public in general recognize that medications with positive effects on mood, and we've talked about some of them earlier this morning, and even have misuse potential. For example, things like methylphenidate for ADHD can be used safely and can have an important role in treatments. Okay, let's go to this final one. And this in some ways is one of the most important and also the most frustrating. Misinformation. So, for example, our city IRB still refuses to review our psilocybin protocol, as their initial response and their continued response is that psilocybin is illegal and that any use, medical research or otherwise, is illegal. So there's kind of a fundamental lack of understanding about the whole process of having the FDA give you an investigation of new drug permission and with DEA oversight that allows you to do very legal Schedule I research. But there's a lack of understanding about that that really is a barrier, because we, for example, want to be able to contact the communities programs directly, some of which have city funding. And if the city doesn't review the protocol, we'll be blocked in that. We can run this study, and we are on Penn territory at the Penn Hospital, but it would sure be nice to be able to reach out in the community, which is what we always plan to do, to reach as many people as possible to be beneficial. But we're blocked in that. So how to get around that barrier? Well, I think, public forums, obviously continuing to write papers, panel discussions, interviews, media, all highlighting the importance and the legality of psilocybin research is the critical thing here. And I do really think that forums such as the Philadelic Forum this morning are really critical in getting this information out and keeping it in front of people so that they understand that it's not only critical to do this research, but that it's absolutely legal. And I'll just quickly say here at the end that for many of you now who are inspired to run out and start some research, the number of months that it takes to actually get there from the time that you actually start, write a protocol, or even get funding can be a little daunting. Dr. Guziak in our group is doing a survey study from people who are investigators, asking them, what was the number of months that it took you to get through the IRB, to get an investigation of new drug permission from the FDA, or to get DEA processes through and getting to obtain the meds. And you can see that if you add all that up, the average over there is about 2 years. So I guess by that metric, we're still in good shape, but barely. We, have gotten through all of our processes, and we dosed our first psilocybin patient just before Halloween. But that was about 20 months from the time of the grant awards. But I hope that all this will be inspiring rather than daunting, and that more and more of you will take on the goal of psilocybin research. And I think that's it, but I would love to hear comments or thoughts about ways of getting around some of these barriers.
Thank you so much, Professor Childress. It always blows my mind how many things you've had to go through for so long and how you've persisted in the face of all of those. It's incredible. Thank you.
You're very welcome.